Yeah, it's obviously my NC State.
And I'll introduce you briefly.
Yep, haven't heard.
I would ping her. Just to confirm, who is that?
DEMING. I wrote her that.
Yeah, I saw that.
Tuesday or so. And she said she had everything on her calendar,
All right.
but I never heard back from the dissertation.
Oh, she wouldn't be reading the dissertation.
Okay.
Okay, as long as she said it's on her calendar, that's good. All right, excellent. No, always good to remind us.
This,
Right up there. Yeah, there you go.
Are you sharing your slide yet? I think you stopped your sharing earlier.
I actually didn't want to get on this thing because there.
through there.
Yeah, it was down there in the lower left-hand corner. I think it was kind of showing itself there. There you go. Turn on. Yeah, that's perfect. Yeah, show you, baby. Well, that's, would you please, since you have Zoom up already, just monitor the chat with the,
Yeah.
Recording in progress.
so, oh, somebody's got a, all right. Yeah, we can, yeah. Okay, no worries. I probably,
Dr. Brady.
when I get some back in here, I'm just.
Sorting.
But, we're gonna apply for that sooner rather than later. I've been told by Carl, your channel. And we're okay. Talk to you. Just give me. Yeah, no, it's, yeah, I figured it's- you would have these as the process. Certainly, that's how you follow. I don't know that I want to be there. Follow on. It does take an interview. If you're interested.
Or too many people here, I know.
Let's see it. Yeah. Well, yeah.
No, no.
No, but it's scary overall. So. Yeah.
This is great. Right.
We're doing this.
Gang, before we get started, what I'd like to say is as you're coming in, please grab an evaluation sheet. For those of our team, this is your chance to rough them up a little bit more. Yeah, there you go. This is an evaluation sheet and just bring them in at the end of the seminar, please. We give those to our graduates, to the graduate degree, and for evaluation purposes.
There's only one left.
That's yours.
There it is.
We got enough, I guess.
Right.
Only one left.
You wanna make copies?
Probably.
I think it's.
Get a roll back.
Sharon.
All right.
Let me know. Ask you what you don't get. This right here is there. Oh, yeah. It's okay if I'm sitting
I love it.
An Aussie.
Start running on time here.
I don't think I will be in the summer, just say, let things change out. We really can.
I haven't seen the crowd like this before.
Yeah, you should have charged.
I thought we're all getting paid for this.
Should have sold picture.
Carlos, you have a key, use the key to the door.
No, I don't.
I used to have one.
I have. Do you want to unlock?
Yeah, I want to unlock it so I can close it so people can come in late. Do you have keys?
No, I thought. Oh, yeah, perfect. Wow.
Yeah, yeah, well, that's. Yeah, yeah, that's great. That's a good lot. Yeah, that's too
I'm ready to evaluate. So you can get the value.
Boy, if I could get your attention, boy, when it turned out this morning, huh? I want to thank you all for coming. I just want to say it's a really special occasion for me to be back here today. And I'm really, really happy to be here to be introducing Simon for his PhD seminar and then shortly after his defense. I think he said something like, all you are invited for beers after. But I'll let you keep that later on. All right, before I begin, I want to tell you a really quick story. And then I'm going to introduce Simon and I can get out of his way. And it's kind of an interesting story and there might be a little bit of lesson in it. I'm still trying to figure that lesson out. About 6 years ago, one of my childhood friends, who I go all the way back to middle school with, we had a lot of adventures over the years, called me from Oregon. And he told me that he had met a young fella out at Oregon State University in Corvallis that was interested in plant breeding. And he was interested in relocating for various reasons to Raleigh, North Carolina. And when I considered him, taking him on as one of our staff in plant breeding or consider him as a student. And same as Mike Sukop, Mike might be on Zoom because I gave him the link. We go back a long, long ways. He can tell you lots of great stories. But Mike said, you know what? I think he's a goodie. And I'd really consider him. Mike's a faculty member down at Florida International University as a hydrologist. And we've been tight for many, many years and we've kept in good touch. And I said, sure, Mike, definitely. And Simon contacted me shortly thereafter. We agreed to bring him on as a summer research technician in our program. And he did so well that we immediately took him on as a master's. He did so well, we agreed to keep him on for his PhD. And it was a really good decision that I made. He's made us a better breeding team. He's worked well with all of our staff and a lot of his colleagues in this room. And the turnout is part of the reason as why he's done so well and how much of a team player he is. And I want to thank him for bringing so much to our program. And I'm looking forward to working with him in the future and seeing where his career takes. So I'm not just stealing any more of his thunder. I'm going to let him get going here with his talk. It's all yours.
Thank you, Craig. And let me just say, this is the best attendance I've seen in this room. Oh, graduate student Modesta Bugu has her seminar on March 17th. Sorry, Modesta.
I expect a big turnout from that side too.
Yeah, and we'll start charging entrance fees, I think.
Or at least an unrestricted gift when you leave the room. Go ahead.
So today I'm going to be talking about my PhD work and a little bit of my master's work in developing genomic tools for sweet potato variety development. This includes genomic selection, fusarium and root knot nematode resistance, and a high throughput phenotyping strategy for nematodes. So I grew up in Olympia, Washington on the Puget Sound, and my first job in agriculture was on an oyster and gooey duck farm. It was at that time I realized I wanted to go to college for agriculture. I went to Oregon State, as Craig mentioned, and then I came here for my master's in agriculture. Today I'm going to talk about the sweet potato. In North Carolina, the breeding and genetics of sweet potato. I'm going to talk about root knot nematodes, which are one of our biggest problems right now. including marker-assisted selection and a high-throughput phenotyping tool. Phenotyping is measuring a trait. Fusarium wilt disease, another one of our big pathogens, and some progress we have towards marker-assisted selection. And lastly, I'll talk about genomic selection. Let's start in North Carolina. What we're looking at here are first year sweet potato seedlings in the field. We're going to walk these fields and make our selections. And as we make selections, we're always keeping this in mind. These are sort of our idiotypes. Covington is the major sweet potato variety released by Dr. Gancho and Ken Pacoda in 2008. This composes 90% of North Carolina's acreage today. It's the major variety in the southeast. But other varieties like Beauregard and Bayou Bell are sort of in a similar product profile. So as I'm talking about idiotypes and our goals, this is what we're trying to meet or beat. North Carolina produces about 62% of the US crop. That was in 2023, worth around $300 billion on an annual basis. And here we are planting sweet potatoes in the field. We're using a clip planter, and really sweet potatoes are clonally propagated. They're just a little, about a foot long slip or stem vine cutting. Those go into the soil with a little bit of water, and it's amazing what they can produce. And when it comes time to harvest, we walk the fields. You saw those first year materials. And really that first year, we make one decision, keep or cull. But keep in mind that while that is a binary decision, there's a lot of things that we're observing in the field. Ken, our field breeder, is making dozens of observations, whether he's conscious of it or not, working towards these product profiles. And of course, there's things that stand out and are easy to get rid of, like cracks, pore shapes, no yield, etc. Let's talk about the breeding program. So this is a sweet potato breeding cycle as I understand it. We produce seed from paired crosses, so we know both parents, or from polycross nurseries, where we just know one pollen comes from insects from neighboring plants. And we typically will plant around 20 to 50,000 seedlings, unique seedlings, in 5 hills in a single location. Each year, we make selections and we whittle this population down. And as we have fewer and fewer lines to evaluate, we can apply more resources to them and evaluate them in more locations. And in a complete breeding cycle, hopefully we have a variety at the end. That takes about, let's use this laser here, that takes about 5 to 8 years. But realistically, the breeding cycle is how long it takes to go from the seed, more likely you're going to become a parent the next breeding cycle. In subsequent years after that first year of keep or cull, we then make more detailed observations. And each year we evaluate more and more traits. In years 2 and 3, we're going to use our field book app, and we're going to rate about a dozen traits in the field using the International Potato Center's descriptors or modified versions of those. As we progress in the breeding cycle further, now we have fewer lines and we can evaluate them for more costly, we can conduct more costly phenotypic assays. These are the 4 major diseases. This is pre-harvest now of sweet potato. The guava and southern root knot nematodes, Fusarium wilt disease, and soil pox. I'm not going to be talking about soil pox today, but the other 3 we will address. Let's start with root knot nematodes. This is the Meloidogyne genus. There's over 100 species in this genus. The ones we're most concerned with are, again, are guava and southern root knot nematodes. And most of this is going to be focused on guava. Guava is a new pathogen in North Carolina. It's been here since, I think, 2011, and there was an internal quarantine on it imposed. So North Carolina produces about 62% of the US crop. 40% of our crop goes to Europe. So with this internal quarantine, we can't have roots leaving the state. This has been devastating for farmers if they have guava in their field. Guava root knot nematode is a plant parasitic roundworm, and it causes these characteristic galls on the roots that make it look like the roots are tied in little knots. Unfortunately, we have just one major commercial variety, I should say modern commercial variety, and that's pictured here in purple. That's Murasaki 29. It's pretty far removed from our sort of idiotype that we're working towards. And it's got a number of other horticultural issues that don't make it a great parent for guava resistance in an orange breeding background. Guava root knot nematode is worldwide in its distribution, mostly in the tropics. And as I mentioned, it's been in North Carolina since 2011, the US since the early 2010s. Recently, this is a figure on the top right from Dr. Adrian Gorney, our nematologist at NC State. And this is updated as of this year. So there's been at least one field detected, guava has been detected in each of those counties in dark red. And you can see it's right in the heart of sweet potato production. Sweet potato is a particular problem, although this nematode can parasitize many other crop plants and weeds and native species. Sweet potato, because it's in the soil, when you move it into a new field, it's easy to propagate this nematode as you propagate the sweet potato. So we started by looking at a biparental mapping population. This is a cross between an African landrace that's resistant to root knot nematodes, a Tanzania, and Beauregard, which I mentioned earlier, is susceptible to the nematodes, both species. And one thing that's cool with sweet potato, they're highly heterozygous, and when you cross them, you can get segregation at the F1 level. What that means is you can visualize it here in these colorful images. These are all TB, Tanzania Beauregard progenies. Well, what we can see in this trait, imagine that's also happening with disease resistance. You have your resistant parent, your susceptible parent, and then everything in between. We measure nematode resistance typically using what's called reproductive factor. This is the ratio of eggs that go into a trial. I extract the eggs and I count them at the end. If you have more than you started with, you're susceptible. If you have less, you're resistant. And what we saw was, so a reproductive factor of 1 would be having as many as you started with. And we saw that approximately half this population was susceptible and approximately half was resistant. And really, a fairly high level of resistance here, about 45% of the population had a quarter of the nematode we started with. This was exciting to us as breeders because despite being a hexaploid with 6 copies of each chromosome, a major gene could explain this phenomenon. And so we did what's called a quantitative trait locus analysis. This is essentially doing a statistical test at a bunch of different marker positions along the sweet potato genome and comparing that to our phenotypic data, that trait data we just looked at. And on the x-axis, you have markers. LG is a linkage group or chromosome. On the y-axis, you have LOD. This is sort of think of it as inversely related to your p-value. So the smaller your p-value, the more significant those markers are in influencing your trait, and the bigger spike you're going to have. In this case, we had a major QTL on chromosome 4, explaining about 70% of the variance for resistance to root 12 or root knot nematode. This was really exciting for us. This was the first look at the potential for marker-assisted selection for this problem. But my work was not unprecedented. Dr. Bonnie Aloka, who's with us here today, when he did his PhD in our program, he worked with the southern root knot nematode. Not as economically concerning as guava, although it could be argued that it is. Guava's newer, more scary. But any variety release that we make needs to have resistance to both. Covington is resistant. We do have resistant backgrounds. But Bonnie identified a major QTL on chromosome 7, explaining about 60% of the variance for resistance. Another really good target for marker-assisted selection. And we developed what are called competitive allele-specific PCR markers. So most of us are familiar with PCR tests. You introduce a primer, and it has a dye on it. And if a certain DNA fragment is present in a given sample, it's going to amplify and fluoresce. You'll see more of that dye. So each dot on these CASP cluster plots, let's look over here on the right. Each dot is a TB individual from that population, and these all genetically resembled Tanzania at this marker position. Conversely, these all genetically resemble Beauregard. So that's all well and good. Now we need to go back to our raw phenotypic data and ask the question, are these markers predicting phenotype? And the answer was yes in a few cases. We have 3 markers now for guava that are between 90 and 96% accurate at predicting resistance. For southern root knot nematode, we have 1 that's between 80 and 86% accurate. This was really exciting, and we have since deployed marker-assisted selection in what we think might be the first time in this crop. This was 2023. I took second-year materials. I sprouted them in the greenhouse, so they've been selected in the field twice now. Sprouted them in the greenhouse, took a DNA sample, sent it to a lab, and we know a month later which of these are resistant to nematodes, which are not, without ever inoculating them with nematodes. That saves a lot of money. But we're never going to release a variety without actually putting it under real nematode pressure. And so we still need to phenotype, and there's still a need to make that a little bit more efficient. I got one of these counting slides. This is a veterinary counting slide for counting a fecal nematode sample in horses. But it's very similar to a hemocytometer for counting blood cells and spores. Same logic applies here. I can count cells within this grid and extrapolate it to a larger volume. So I extract a 50 milliliter sample of nematodes in water. I take out a subsample of that and I count those eggs. It stands to reason that I could take a subsample of the subsample and extrapolate about the whole. So that's what we're doing on this slide. What I did was within that grid, I just counted a small but perhaps representative number of seconds, 2, 4, 6, sorry, 2, 4, 8, 12, 24. What you're looking at here, this is called a Pearson parallelogram. So on this diagonal, we have distribution plots for how many eggs were present in each count. In this top sort of triangle here, you have the correlation between the given counts. So for instance, 2 cells and all 48 cells had a correlation coefficient, or R, of 0.89. That's pretty good. I decided somewhat arbitrarily that 8 cells was probably the best bet for us. It felt like enough, and it was representative, and we get diminishing returns while counting. It's more work after that. So this was one effort to increase our rate of phenotyping. I also put a camera on the microscope because it's much easier to look at a screen than go cross-eyed staring at a microscope all day. And so I started collecting images and further put a smaller grid on my computer screen so that I was able to count the eggs in a simpler fashion. It was one day on Friday that I was doing exactly that in the lab. And my master's student at the time, Mark Watson, he's a PhD at UC Davis now. He comes in and he's giving me an update on his project. He works with ornamental sweet potatoes. And one of his projects was to use machine learning and image analysis to measure sweet potato leaf shape. And leaf shapes in sweet potato can be heart-shaped, willowy, lobe. There's so many different patterns. And I said, Mark, that's really complicated. You think you could count nematode eggs like that? And he pauses, which is a mark of genius, by the way. And he's like, yeah, I could come up with something. So that was Friday at like 5 p.m. Monday morning, we had the first iteration of our machine learning approach for counting nematodes. I'm going to talk about 2 approaches here and then a hybrid approach. So the first uses what's called a convolutional neural network. This sort of emulates how people think. You recognize a pattern, and within that pattern, you can further recognize smaller and smaller or larger and larger patterns and make decisions about images of the whole. The first step is to binderize the image, turn it black and white, and then just look at all the white regions and try to decide what kind of object those are. In this case, we start with a training set of data. We start with pictures of pictures of 1 egg down here in green boxes, pictures of 2 eggs, because this object would look like an L, not 2 individual pill shapes, and then debris, cellulose, sand, that kind of thing. And it worked pretty good. I'll point out a couple spots here. So like there's 1 egg right here that did not get counted. Here's a double egg that got counted as 1. So it had its problems. I just switched slides there. This is a second approach to contour method. This does not use machine learning. This is human defined parameters that Mark and I kind of decided on our own. And we look at things like the area, the number of pixels of that white box or that white shape, the length, the width, the ratio between those 2 tends to be a set number, curvature of the eggs, etc. And this did a pretty good job. Like you can see here, it did detect that one the other approach missed. So, but it still has its faults. It didn't catch this one, this double egg over here. And so we decided, let's combine the 2 approaches and add a few more bells and whistles. And we ended up coming up with an approach that works quite well. What we're looking at here are 3 nematode species. I've introduced guava and southern root knot nematode, Lodogen javanica. This is from a project that a master's student, Savannah Moore, was doing. And I asked if I could use her images for this publication. She said yes. Again, we have another Pearson parallelogram. We're going to see a lot of these today. So if you don't understand how to read these, please tell me now and I'll pause and explain it again. On this diagonal here, we have our visual counts, convolutional neural network, our contour, those are the human defined parameters, and then our hybrid approach. This is a new one on this slide. And really the hybrid approach just considers the best of both worlds. Using this hybrid approach, we had a really tight spread on our scatter plots, and it was very highly correlated with our visual counts. I've never actually seen an R of 1 before, so that was really exciting to see. But this is a fairly simple use of machine learning. As I mentioned, we published this paper last year. So that pretty well puts a handle on root knot nematodes for us. On the breeding side, on the phenotyping and genotyping side, it's still going to need to be a holistic approach to tackling this problem, including cultural management, chemistries, extension work, etc. Now let's talk about Fusarium. Fusarium wilt's a fungal ascomycete disease. Fusarium oxysporum can affect many plants, and there's, I think, over 100 species. This is, we're gonna be talking about conidia today, micro and macro conidia. These are the types of spores that we're gonna infect sweet potatoes with. And in nature, in the field, these are gonna germinate, a hyphae will grow up and penetrate into the plant's roots. And then they'll colonize and clog the vascular tissue. So even if there's plenty of water in the field, you can have these wilting symptoms on the top of the plant. With sweet potato being planted from cuttings, you're not going out with roots, you're going out with a wound, an open wound in the field. And that's one of Fusarium's favorite spots to colonize. So here's what it looks like in sweet potato. Again, Fusarium oxysporum, Forma specialis patatus. This is a host plant specific designation. So there's many different Forma specialis. Field symptoms include yellow leaves, stunted growth, discoloration of the vascular tissue. That's how we're going to diagnose it in a second. And of course, mortality, reduced stand count. Total crop failure has been reported due to this disease. But I should say here that most of our growers are not really aware of it. It's not a problem, and that's thanks to cultivar resistance. I like this quote from Chris Clark, the LSU pathologist who recently retired. The most effective and efficient means of managing Fusarium wilt is the use of resistant cultivars. We've done such a good job with that. By we, I mean the whole sweet potato breeding community, that our growers aren't concerned. But if we're going to release a variety to replace Covington, Covington has this resistance. So necessarily any new variety will need that as well. I worked with a new mapping population, DMO4-001 by Covington. It's quite a bit larger than the TB population. DMO4 is susceptible to Fusarium wilt disease. Covington is highly resistant. So once again, we expect to see segregation of the disease, just like we do for skin color. I use an ordinal rating scale from 0 to 4 to evaluate symptoms. First, we set our trials up. I cut my plants. These were in a greenhouse that was clean, virus-free, no other diseases present. Cut my plants, let them wilt just a little bit overnight, and the next morning I dipped them in a spore solution. Letting them wilt causes transpiration to kind of accelerate, and they uptake those spores, and we're hoping that it colonizes the vascular tissue. In this case, we cut the vascular tissue at about a 30-degree angle with a razor blade, and we looked for symptoms of signs of discoloration. In this case, Covington, our resistant parent, was a 4, so asymptomatic, and DMO4 was a 1, close to death. These bioassays are run between 2 and 3 weeks, depending on how our check lines perform and the parents' known lines. And then we evaluate on a scale from 0 to 4, and we conducted trials in 2017, 18, and 2023. These are done in the greenhouse in little plastic 4-inch pots, and each pot is a biological replicate with 5 copies of a given DC genotype in each. So let's look at our check performance first, because this is how we gauge how well the assay went. So in 2017 and 18, a master's student, John Kinchick, conducted these studies before my time. One thing that stood out initially was that we did not see very good separation of the parents in that first trial. That's a little bit concerning. We want to see DMO4 getting hammered and Covington looking pretty good. So he opted to repeat that trial again. Now you see much better separation between the parents. And again, we're looking at a 0 to 4 rating scale. So 0 is highly susceptible. I came and started my PhD in 2022, and I wanted to repeat this trial and turn it into markers that we could use for selection. Once again, checks performed pretty much as expected, and Covington was highly resistant, DMO4 highly susceptible. Here's a distribution of our phenotypic data. So again, 0 to 4 scales on both axes. And what we can see is that there's a pretty normal distribution, which could be indicative of a major gene, although that doesn't necessarily tell us that. We can see that between trials, the correlations are pretty good. Now, I don't expect it to be as good as the nematode egg counting, because this is 2 interacting biological organisms, trials conducted by different scientists in different years. I'm pretty pleased with how well correlated these were. And so then we did what's called a joint analysis. I use linear mixed models for that. If you're curious, I can tell you more after the presentation. And once again, we did a quantitative trait locus analysis. Now I skimmed over the last one because I talked about that in my master's seminar. But you need 2 things to do this. You need phenotypic data, which you just saw, and you need genomic data. And we're going to do statistical tests at every marker position. Well, when we genotype, for this population, we use GBS, genotyping by sequencing data, and you have a bunch of puzzle pieces, if you will. We need to figure out how to assemble those. And in this situation, we didn't have a final picture of the sweet potato genome, but we had what I think of as like a reduced resolution image. We have a relative a diploid relative that's simpler in genomic structure, but has highly conserved genes with sweet potato. We use that as a template or a blueprint to construct our genome, and then we perform the statistical test. And that template was constructed by using MapPoly, which was co-developed by Dr. Molinari here. Once we had the linkage map, we did our statistical tests. We see that there's very clearly one major QTL on chromosome 10 that was detected in all trials and in the joint analysis. I might add it was detected in each replicate individually as well, so very consistent. We also have some minor QTL. We didn't see these on the nematode graph, but there's some that are maybe just a little bit at or above the log threshold, typically around 4 or 5. These certainly do appear to be contributing to resistance, but not consistently so. I didn't see it in 2023. It did pop up in the joint analysis. And likewise, I found another minor QTL here in chromosome 6 that was not detected by John or in the joint analysis. So really, let's just focus on the big one. And where we're currently at is we've identified candidate genes. So we use a blast search. This is called a basic, I think it's local alignment search tool. Basically, I go and look at the Ipomyotryphyta reference genome, and it has a bunch of annotated genes, and I can look at each gene in my QTL confidence interval and search the literature, see if those genes in other crops are associated with fusarium resistance. We found 404 genes, so that's a pretty big literature search. But if we just look at the QTL peak position, where most of the variance is being explained, I found 7 candidate genes. And these genes are, for the most part, in the NLR, nucleotide leucine-rich, nucleotide binding leucine-rich gene family. And these are associated with effector triggered immunity. This is one of the innate plant immune system responses. So the plant will recognize molecules excreted or secreted by the pathogen, and it'll trigger an immune response. There's a lot of different modes of action. These candidate genes were in chickpea, melon, tomato, soybean. We looked at the Arabidopsis genome, and actually just recently we published a hexaploid sweet potato. No, we didn't, but the community published a hexaploid sweet potato reference genome, and I was able to scour that for reference genes as well. These 7 genes will be priority targets for marker-assisted selection this year. And we just, I'm pretty sure we just got funding for that, Ken, that meeting yesterday. So now let's transition to genomic selection. This is a really difficult concept to wrap my head around, and I'm still learning, of course. So I think we understand marker-assisted selection at this point. Marker-assisted selection is looking at maybe just one position in the genome, and we know it's associated with a trait of interest, so we can select for the gene and make that decision much earlier than having the phenotype. Genomic selection is founded on similar principles, but instead of looking at single markers, we're working with traits that are more quantitative in nature. So they're controlled by many genes spread throughout the genome. The other, I guess, key difference from marker-assisted selection and phenotypic selection is that genomic selection uses these marker predictions not to select the best individuals, but to select the best individuals based on genetic merit. So it might not be the best looking thing in the field, but it has the potential to be the best parent in the field. So it's a bit of a paradigm shift and a deviation from phenotypic selection, which is how plant breeding has been done for all of human history to this point. I think to talk about genomic selection, we need to understand the breeder's equation. There's a bunch of different forms of this. This one's pretty common in the literature. And what this says is that delta G, the rate of genetic gain, is equal to selection intensity times selection accuracy times our additive genetic standard deviation, all divided by breeding cycle length. I think cycle length is the most intuitive one here, right? If we can shorten our breeding cycle by using markers and making those predictions earlier, let's say we go from evaluating traits in year 6 to predicting with markers in year 3. We have our breeding cycle. And so in theory, we doubled our genetic gain. Speed breeding is doing a lot of research into this, ways to cut down on the breeding cycle length. Let's look at selection intensity. So this is how much your selections, how far beyond the population mean they are. Are you selecting only the elite most 5% in the field? Your selection intensity is really high. Genomic selection doesn't directly influence this, but as you make better selections with genomic selection, your selection intensity could improve. Selection accuracy is highly influenced by genomic selection. This is probably where we have the most to gain, but it's a little less intuitive than breeding cycle length. Selection accuracy says it is a measure of how close your predicted values are to the true breeding values. If we're using markers to predict, we get a lot closer to the true breeding value than if we use phenotype to predict, because phenotype, while it is an expression of genes, it doesn't directly correlate to what's in the genome. So now we're shifting to using the genome for selection. I'll say additive genetic standard deviation is an aspect of the breeding population. It's not influenced by genomic selection. But if you want to change this, you could add new and diverse individuals. So in order to do genomic selection, you need what's called a training population. We're going to use this to train our predictive model, and then we're going to test it out in an actual population. So this training population, conceived in 2018, initially meant to address guava root knot nematode, but we put a handle on that pretty well. So then I switched to focusing on more horticultural agronomic traits like yield. Fortunately, each of these families, so there's 33 families. This is a polycross nursery outdoors. So we'll graft onto a Brazilian morning glory, let the plants grow up and trellis them. And then this morning glory doesn't put energy into storage roots. It's just got, normal fine fibrous plant roots. And so the sweet potato can't put energy into the ground. It's going to put energy now into seeds. And it produces lots and lots of flowers, and bees tend to pollinate these, and we go collect the seed. So I know who mom is. I have no idea who dad is. It could be anyone in this checkerboard nursery here. So keep that in mind when I start talking about pedigrees. We don't know dad in many of these cases. And further, many of our lines are produced in this fashion historically. So most of our pedigrees, historical pedigrees, are half complete. We planned it out in the field in 2022, our training population. This was 3 locations, and it was a single replicate per location. For context, these are like 1 acre trials, tremendous amount of work to handle these. And we didn't have a ton of plant material because they had just been seeds a couple years prior. Population size was around 650. And then we also evaluated a breeding population. This is our validation data set to make sure our model is actually doing a pretty good job. This was a more selected, more composed population because it was represented by most of our elite breeding germplasm. Whereas the training population is a cross between elite stuff and guava root knot nematode stuff. That tended to be not as horticulturally refined. Everything was planted in 5 plant plots in the field using conventional production methods. And to phenotype, we used our Exeter AccuVision optical grading system. Here's Craig standing at it. Essentially what you do is on one end, you dump in a bucket of sweet potatoes, and it goes up a riser belt and then onto several belts that are increasingly fast. What this does is simulate the roots from one another, passes beneath a camera, and we get pictures of each root. There's a black box software package in there that measures or estimates the yield on a root level basis. And then I can compile those into plot level, and I can do my analysis. The traits that this measures tons and tons of parameters. The ones we're most interested are total yield, length to diameter ratio. This is our best, easiest proxy for shape, maybe not best. Number of roots produced, and USDA grade, specifically pack out, which is what growers refer to the USDA #1 storage root as. So this is where they make most of their money, and that's what we want to be selecting for. Let's look at data. So this is the distribution of phenotypes in both populations for the 4 traits that I mentioned. I don't want to dwell on this too much, but it's possible to do indirect selection where you select for one trait early that's highly correlated with another one later on. That's not really going to come into play here. But this does show us some patterns that we've observed in the past. So for instance, the more roots you have, the higher your total weight is. It's a pretty good correlation. That's something we've seen in the past. And likewise, pack out is highly correlated with total weight. And that makes sense because growers are making most of their money off high pack out. So that's going to be what most of total weight is composed of. We're looking at the average performance for each trait now in the training and breeding population. So first thing I notice, length to diameter ratio is lower in the breeding population. Well, this is a more refined population and we've imposed selection bias here. Our idiotype is between like 1.75 and 2.25, so the breeding population is closer to that target than the less refined training population. Root count, at first I was kind of surprised to see this was lower in the breeding population, but I'll explain. I think the other two traits kind of explain why that is. The breeding population had quite a bit more total weight. It also had quite a bit more US #1 weight or pack out. And so what this is telling us here, root count, is that the breeding population was making fewer, better roots. So we have our phenotypic data, now we need genomic data. And we used this new genotyping platform that is called Diversity Arrays Technologies, or DARTAG. It's a 3,000 single nucleotide polymorphism or SNP panel. And I think most of us are aware, but SNPs are single nucleotide polymorphism. So if sequence is CAT, a SNP might be CTT. This is not a figure lifted from this publication, although it does resemble one in there. This is actually our population, and it shows us where markers are located throughout the genome. All this is to say they're genome-wide, which we need for genome-wide predictions. And this is not a super deep pit. 3,000 markers is not very much. The GBS data I talked about before had like 30 million reads. These are vetted markers, though. These are markers that are known to be polymorphic in diverse world germplasm. And this panel was developed specifically for sweet potato for projects like this. The other thing that stands out, I guess, is that there's some regions that are really enriched for, that have a really high marker density. Chromosomes 13 and 5 have a lot of genic regions on them, which means there's a lot more explanatory markers in those regions. So all this looks normal. This is what we expected, and it was good to see. These are kind of difficult to visualize, and there are, on each axis, we have every individual represented. So there's 700 lines. I don't have a convenient way to show you guys that information, but we'll zoom in and look at a couple spots. But first, I just want to say this is a pedigree relationship matrix. So on the y-axis, you have a list of all your clones. On the x-axis, you have that same list ordered the same way. And what it's reporting is the correlation, the coefficient of relationship between individuals. So it stands to reason that an individual should be highly correlated with itself. On paper, with a pedigree, it's 100%, or a correlation, a coefficient of relationship of 1. Genomic relationship information can tell us a different story. So, looking at the pedigree matrix, we have parents Monaco and L1431, and then all these ML lines are biparental full siblings of that cross. Our expectation is that full siblings share half of their genetic information. And that's why it's all shaded in the same tone of orange. This would correspond, if there was a legend here, this would correspond to a coefficient of relationship of 0.5. And then of course, 1 on the diagonal. Monaco is 100%, shares 100% of its genetic information with itself. But when we look at the marker data, we realize that we can have deviations from this expected shared half of your genetic material. You could have 0.51 or 0.49, and we can determine that using marker information. We can also notice if there's a pedigree mistake, which does happen. ML496 does not correlate very nicely with any of these in either dimension. So there's a good chance that this was just a mix-up, clerical error, maybe the wrong pollen got brought to that flower, and I can pull him out of the population. Also, this little box down here shows us that these two individuals might be the same. And ML174, ML194, I've certainly written 9s that look like 7s before, so that could partially explain that. Now we're going to look at a principal component analysis. This is also kind of a tricky thing to wrap your head around. So what we're doing is looking at sets of markers that explain the most variance in these populations. We want to, so if we start out, we have 600-700 individuals, 3,000 markers. That's a giant matrix. There's not really a good way to show you guys that information. But we could imagine that each individual is a point in 3,100 dimensional space, and we could draw a line from that point to other points, maximizing the distance from that line to our other individuals. In other words, we can draw this imaginary line through many dimensional space that explains the most variance in the population. And that variance, we could think of this as sort of like a pseudo marker. It's a marker representing all considered data. What's nice with PCAs is we've already considered all the data, but we've reduced the dimensionality to 2 so I can show it. All that's kind of a mouthful. What does it mean? Well, let's start with a simple part here. ML, we were just talking about these individuals, a biparental population, nicely clustered. What that means is that their markers are largely shared and they're closely related to one another. I also see that ML 498 is not clustered with the family. That's what we saw in the genomic relationship matrix as well. I think a broader and more important observation here, though, is look at our training population, our red dots, and look at NCSU. This is our entire breeding germplasm. The training population has a similar pattern and distribution as our breeding population, which means that our training population is representative of that population. And in theory, our genomic selection model could work. Now we need to talk about blocks and blues. Again, this is a tricky concept to understand, and I'm not claiming to have a perfect understanding now. But they're used for a couple of reasons. These are estimates or predictions, and they're the best in that they minimize variance in predictors. They're linear because they're composed of linear mixed models. I'll show you what that means. They're unbiased because they, let's see if I can remember it this time. They're unbiased because they, the expected, the estimates on average should be equal to the true breeding value or true value that we're interested in. And then, of course, they are estimates or predictions. So again, what does this mean? Blues use fixed effects. These are factors we can manipulate or control in our experiment. Block, row, column, things like this. You might consider using genotype as a fixed effect. If I'm doing a nitrogen rate study like Schultheis's team does, and I want to know how Beauregard and Covington do, I would treat these genotypes as fixed effects because I'm specifically interested in those genotypes. Random effects are when we want to look at, say, a set of genotypes and generalize about another population. These would be predictions, and this is where we use blocks. So random effects are not directly controlled. Genotype in this case could be a random effect. And so it's confusing in class when you see genotype fixed in one session and random in another. This is part of the reason why. I'm going to show you both instances. And the reason that I use genotype as both fixed and random is because I do a two-stage model. The first is I'll show you on the next slide, but I do want to bring up this concept of shrinkage. Shrinkage is basically a little weighting factor that reduces the impact of outliers on your data. So if you're kind of close to the population mean and you consistently have that trait evaluated, this isn't going to affect you very much. But if you're an extreme outlier in one rep and not the other, shrinkage is going to reduce the impact of that on our generalizations. So our first model uses fixed effects. A genotype is a fixed effect. YIJK is a vector of phenotypic observations for this clone and that trial and that block. Our model intercept is just the population mean for a given trait. So we would run one of these for each trait, really hundreds of these for each trait. Fixed effect for clone, and then a fixed effect for block J and clone I. Now we use an augmented row column design. What that means is doubly resolvable augmented row column, but that's a mouthful. What it means is that we've included checks, these little red pluses, exactly once each. Covington and Beauregard appear exactly once each in each block. This allows us to use Covington and Beauregard performance to estimate our block effect. So this is how blocking was estimated, was using our checks. Those are the only genotypes that are repeated in these studies. Every other black plus is a unique individual. And then of course we have our error variance. This is not explained by the model, so it could be weather, any other experimental parameters that we haven't accounted for. Model 1 uses clone as a fixed effect. Model 2 is much simpler. Now we've already accounted for our fixed effects. Stage 2, random effect for genotype. The reason we treat it as a fixed effect in the first instance is to avoid double shrinkage. So shrinkage is only occurring once in this scenario. And really, those were linear forms and talking about vectors, but all this is matrix algebra. I don't claim to understand how to do this by hand, but this is the general form of the matrix form. And what you have is your vector of observed phenotypes, a matrix and your fixed effects, matrix and your random effects, and error. Let's look specifically at how we incorporate marker data, because we haven't done that yet. Essentially, this U component in the G-BLUP model, which is what we're talking about, says that the variance of U is equal to the relationship matrix times the additive genetic variance. And so we looked at the pedigree matrix, we looked at the genomic relationship matrix, those are those heat maps that we zoomed in on. We're also going to consider, so pedigree is A, genomic relationship is G, and we're also going to consider both data types together, another hybrid approach. This could be beneficial because sometimes you genotype and a few samples have low quality DNA and you don't get anything back. But you might have pedigree information. It could be partial pedigree information, but why not consider all available information? And this is really the brass tacks. This is what the model spits out. This is what Ken or Craig is going to use for making the decision. Let's just look at the number of storage roots. On average, the training population produced 26.75 storage roots per block. After we run our model, it spits out expected or a genomic estimated breeding value for each individual. So from these three, I would want to select 200102 because he produces 5 roots more, and these two are less than the population. So when I add this to these 3 numbers, I get these 3 numbers. And then I could do this in Excel. I could rank them and just pick the best ones. Those are the ones to use in pairs. It's a little more complicated than that because we have to consider lots of different traits, and there's ways to index and weight them. We're not going to get into that today, but in essence, this is the thought process we went through, and we made some selections and are breeding with them now. Let's talk about how well these models worked. So predictive ability is how well correlated your phenotypes are with your GEBVs, your estimated breeding values. And we're looking at training and breeding populations here again with all 3 matrix approaches. These numbers are R. They're the correlation coefficient between the phenotype and the GEBVs. And admittedly, they're not as high as we'd hoped. But we'll visualize this a little better. One thing I do want to point out here, though, is numerically, in both the training and breeding population, the G matrix approach outperformed the other 2. Here's our predictive abilities plotted as box and whisker plots. And we have our 4 traits on the bottom. Predictive ability, again, unitless. This is just our correlation coefficient. And you can see that on average, the median line there in the center, the G matrix outperformed the other 2. Now we're looking at the training population right now. We'll look at the breeding population next. One thing about the training population that we know is that when we harvested those seed, we just had mom. So you only have half the pedigree information for these individuals. And that could be a big part of why pedigree didn't do as well as the other 2. Now we go to the breeding population, we see pedigree does a little better. Well, in the breeding population, we often know both parents. Often we create these individuals through paired crosses, so the pedigrees are going to be a little bit better. Yes. So how do we tie all this back to our breeder's equation, and how do we turn that into a product, a result? Well, bear in mind that from an outside perspective, you may think our goal is to create new varieties, and it certainly is. But in order to create those varieties, we need to make populations that are enriched for our traits of interest, so that we increase the likelihood of recovering a variety. Back to our breeder's equation. Let's look at time first, breeding cycle length. If we can make our breeding decisions earlier, as I mentioned, maybe in year 2 or 3 with genomic selection, we could have our breeding cycle length for these traits. What I want to see is marker-assisted selection applied in year 2. After a round of Ken's keep or cull decisions, now I have a refined pool here. We look for nematode and fusarium resistance, cull everything else out. These markers are cheap and there's few of them. Genomic selection is expensive, so let's do it on fewer individuals after using these easy filters to get the junk out. We apply genomic selection in year 3, and now we're making decisions about yield and shape, length to diameter ratio in half the time. And further, we're doing it more accurately. And we could potentially increase our selection intensity. So there's a number of ways that these technologies can increase delta G. There was something else I wanted to say here, and it's skipping my mind, but let's continue. So in conclusion, what did we learn about root knot nematodes? Marker-assisted selection has now been deployed in Sweetpotato. When I started my master's, we had 2 NC coded line, so our own creation, 2 lines with guava root knot nematode resistance. Now we have over 120. Oh, actually, I do recall what I was gonna say. We historically have gone from seed to parent in 6 to 8 years. Now we can go from seed to parent. That was the point I wanted.
Doubling genetic gain.
Double genetic gain, and that's just breeding cycle length. We can increase our accuracy and intensity. So yes, we went from 2 to 120, and I expect at least 200 more this year. I'm currently preparing samples to send to the lab for genotyping. We expect this is going to expedite variety release quite a bit. We now have a high throughput phenotyping tool for counting eggs, and that reduces the costs of genotyping. 3 other grad students are now using this counting tool in their projects. So a rising tide lifts all boats. Populations do change though, as root knot nematode resistance is incorporated. For instance, many of these guava root knot nematode resistant lines are really lacking in horticultural and resistance traits to other diseases. So one step forward for guava might mean a few steps back for these other traits. It's really a problem. And lastly, cultivar resistance will be a major factor in management. This is one of my favorite pictures of the year. Right next to each other in a guava root knot nematode infested field, we have NCO 91105. That's one of those 2 lines, by the way, that we had with guava resistance. It doesn't have a single gall on it. Next to it is Covington. I didn't cherry pick. I didn't go find the gall roots and bring them over. These plants were side by side, and you can see not only the yield hit, but you can see the galls on this small Covington root. It's pretty stark contrast. This was true in this particular plot, but I saw this in just about every plot in this field. So just to compound the point, the cultivar resistance is going to be a big part of our management strategy. And that's also going to be true for Fusarium wilt disease. This was the first genomic analysis of Fusarium wilt outside of John's work in 2017 and 18. And we have some pretty promising candidate genes. I think we should use these as targets for marker-assisted selection. That's my goal for the end of this year. I think we do, however, need to explore other isolates of Fusarium wilt. The ones we're using are 40 years old in some cases, and we know that populations change in the field. So we could have resistance overcoming strains out there, and I would be curious to get my hands on those and start to breed for resistance before it becomes a problem. So we don't have the same problem we just had with guava. And finally, with genomic selection, well, I think we can conclude that it's possible, and it should definitely expedite variety release, but our predictive abilities could certainly be improved. Also, implementation of genomic selection, I was talking about when to deploy it, what lines to use it on. We got to figure that out. This is still very new information for us, and we're figuring out how to process it. I'm working with Dr. Vani Aloka on a 2nd-generation training population with a paired cross structure, so we have a better understanding of pedigrees. And I think we also need to explore more traits. I still have all that exeter data. I also have drone data from those GS populations in the field. And I also think this could be an opportunity for us to explore a trait that we expect to be quantitative, like streptomyces resistance, that 4th disease I didn't talk about. What we can conclude from our different approaches, the G-GLUT model did work the best in both populations for all traits. So genomic selection works. The pedigree-based approach had lesser predictions, but we know pedigrees are subject to clerical errors, and the depth of our pedigrees might only go back a couple generations. So it's just not complete information. And the hybrid approach did not outperform the genomic approach, which was a surprise. But I think this could have to do with the quality of our pedigree information. I do want to reiterate that genomic selection is not phenotypic selection, but it complements. Genomic selection is the selection based on genomic merit. It's how good you might be as a parent. So again, you may not be selecting the best things in the field, but using this strategy, we could select the best parents and have the breeding cycle shortened. And lastly, our top GEBVs, our top genomic estimated breeding value lines, are being used currently to breed new sweet potato varieties. And this, we believe, may represent the first application of genomic selection in this crop. And so I've shown this acknowledgement slide in the past. I certainly have a number of folks I want to thank, but let's do it a different way. Here we are on top of the plant sciences building, and there's 3 fellows in this picture that really helped drive these projects forward. Marcelo Molinari, Guy Pereira, and Gabrielle Gestera. So thank you three very, very much. Here I am in the food science pilot plant. We were playing around with a spiralizer, just exploring potential for new product development and generally having fun. We do actually work in there sometimes. Here we are at a field day talking to growers about our new varieties. And on the left there is Mark Watson. He's the guy that I mentioned is at UC Davis now. He helped develop, he really developed the egg counting app. Here I am with the greenhouse crew. These are our pollinators. They make sure that the plants are happy, watered, and maintained. And of course, we get them out in the field as well. Thank you guys so much for your help. Here we are having some more fun in New Orleans, eating sweet potato fries. And I had the best cheesecake of my life at this point. Here we are seeing off our friend Jaron. He's now working with Dr. Gorney's lab on his master's. Congratulations. Here I am actually working for once. That picture is just for you, Russell. I also want to mention the research station staff, the rest of the folks sitting there. We quite literally could not do these projects without their support and help. So thank you guys very much. Here's Craig critiquing my work, perhaps. A little rough at the end of the day. He's a great boss. We are evaluating storage disease symptoms. And this is my favorite picture of all. I had the opportunity to go with Craig to Uganda and meet some of the folks who really pioneered and started a lot of these projects decades ago. Between Craig and I is Dr. Oloka. He's here with us. To Craig's left, from our perspective, Dr. Bernard Yada, Dr. Robert Mwanga, and then Dr. Ruben Sali. And it was really exciting to meet these guys because I'd only seen them on Zoom. I'd read their papers. Dr. Mwanga was a World Food Prize recipient, and he was Craig's first graduate student. So thank you to everyone who's helped out, and I'd be happy to take any questions.
Nice work, Simon. We have some time for questions here. I'd be really interested to hear the audience's QR score.
Great talk. If breeding doesn't work out for you, we'd love to have you in pathology. But I do think you'll be great at breeding. I kind of have two questions, but they're related to the same thing. In your fusarium analysis, first question is kind of simple. How long did you soak your plants in your spore solution? And then to kind of piggyback on that, I noticed that there seemed to be a higher, I guess, like resistance coefficient, if that's how you want to, like the numbers were higher in your trial than the ones that you did not do. Yeah. Do you think that like was the like, what am I trying to think? The inoculation or just the whole protocol changed any from the previous trials to the way you did it? Or do you think there was something else going on as to why any particular line you evaluated and that might have less disease in 2023 than it did in 17 or 18?
Yeah, definitely. I don't recall exactly how long we soaked them for. I think our protocol says 1 minute. Is that right, Ken?
We're sharing.
Sharon? I can get back to you on that. I've got it written down. So, okay, so the question.
Yeah, 1 minute.
1 minute. The question here, as I understand it, is why did I have, or maybe you're thinking of these numbers?
Yes.
Yeah.
So you're wondering why did I have, why did Covington do better and DM4 do worse? And did I see that in the progenies as well?
No, what I mean more is like if you look at like Beauregard, like
Okay.
Beauregard's quite a bit higher in 2023 than it was in 17 or 18.
I see, yeah. It's
Same
All
with Hernandez, really all of them.
all the checks are higher. So back to this explanation here. I ran these assays between 11 and 19 days. I lost my laser pointer, but you can see where that says that. John's assays were longer. He ran them longer. I had a really hot test and my DMO4 plants started to crash. I used that to gauge because I expected that to be more telling of its progenies than the check lines. So you're right, and that's a great observation. But as we see here, the distributions were a little bit skewed towards more resistant in this population, but still more or less centered about 2. This is exactly the reason why we use a joint analysis, where we consider all the experimental design parameters, all the phenotypic observations in tandem, and we can adjust for those year round events.
I can add something because I was a little bit like a witness for both of these. Maybe the cuttings itself. some of the old wood, we've been cutting.
Yeah.
We got older tissues, but the new ones we started to consider a little bit more pressure.
Thank you, Carlos.
Do you suspect that, well, great presentation, so do you suspect that dominance and epistasis are on your way, for getting better predictors?
Absolutely, yeah. I didn't mention that.
That's what happened in cassava application.
Carlos said, why not look at dominance and epistasis? We didn't really have the tools for it at the time. It's much harder to observe. We know additive variance explains most of what's inherited, and these were additive only models. We also didn't really consider dosage. I'm kind of shooting myself in the foot here, but we looked at our most common allele and the most common alternate allele. There's certainly a lot more information here, and all those things could explain why our predictive abilities were as high as they could. Russ?
So a couple of years ago, I think I asked you to put on your plant breeder hat, but now you've got on your plant breeder suit. I mean, that's
I'm not wearing this to the field.
I've got kind of two questions. I think they kind of piggyback. What do you think are three tools or thereabouts that, say, if you were transported to a new breeding program, what are three tools that you would take with you? And then what would maybe be one thing that you think has really good potential but hasn't been worked on enough? And what would you do to work on that?
Okay, I would have to think about that for a while, but just right off the cuff, I would want a reference genome. I want a really good team to help me make it happen. What would be my third?
Money.
Yeah. And then to answer your second question, what do we have a lot to gain from what new technologies and tools? I am really excited to see what we can do with drone phenotyping with Dr. Jing Zhang, Jerome Molesky, Kranos Williams, Mike Kudnow, all professors here at NC State. We have working with the exeter, so much work there. We pick the roots up, we put them on the exeter. There's 5 or 6 people running it. It beats the roots up, so they're no good as seed pieces. If we could phenotype in the field, and we already have preliminary results that are really promising. If we could phenotype in the field, we could forego all of that and make our decisions faster, earlier, and better. So just like with genomic selection, image-based analysis tools have a lot of potential. That's my answer. Modesta?
You had a question in this on Zoom.
Oh, sure. Okay. Let's pull it up. Actually, do you want to just read it so I don't have to find it?
Yeah, it says from HEDOC, says great presentation. Congratulations, Simon. You mentioned that you could increase predictive ability in genomic selection. What would be your approach to that?
I think when I was talking with Carlos a minute ago, I tackled a lot of the things that I would do. I think another sort of way, we're shifting more towards using more paired crosses too. We're getting away from the polycross where we don't know both parents. And so having more improved pedigrees could certainly improve predictive abilities. I think it would also make that H matrix perform just a little bit better than the G matrix. I don't know, I'd like to see it though. And we should probably see that with our new training population I mentioned. This uses an NC2 mating design, HEDOC. If you want to look into that or shoot me an email, I'd be happy to copy one.
Jonathan had his hand up several times here. I saw.
Well, I was just gonna follow up on that Fusarium, the isolate you talked about,
Yes.
40 years old. Were these the same isolates? Were they as virulent? Is that perhaps a cause, or did you use a different isolate?
Yeah, so I actually forgot to mention this point, but when we inoculated, I used a cocktail of all 3 isolates together. I thought I had it on here. We did actually look, I did like a mini QTL analysis. So I just looked at 50 individuals and looked at our 3 isolates individually. Unfortunately, that 50 individuals was not enough resolution to detect any QTL. So if you're doing a QTL analysis, try to get larger populations. But yes, I want to look at, because we use pure isolates because it emulates field conditions where there's multiple pathogen strains potentially present.
But we've used the same isolates.
But we've used them for 40 years, yeah. These came from Chris Clark at LSU, most of them did. And yeah, I think you're right to ask that. I'm a little concerned about it myself. I want to see what's next. And I know Lena's group is doing a really good job. They did a survey in Uganda, and I think there may be some efforts on doing a survey locally as well. I've also asked growers and extension agents to report it if they see it. then I can get my hands on it before it gets its hands on our sweet potatoes. Right.
I had a parallel question, the danger I think breeders have encountered is the boom and bust cycle, right? Because we understand the genomics of the plant, but what do you know about the population structure of the melanodyne, especially the invasive species, the guava root knot, and also the fusarium? Is there good data on effector profiles, population structure, et cetera? Like, what's your level of threat for failure in the future?
So I couldn't comment on effector profiles. I can't talk about population. So guava root knot nematode, global distribution in the tropics. There was a morphological and phylogenetic analysis in I want to say 15, 18, somewhere in there, where they looked at 15 different countries globally and did both morphological and phylogenetic analyses. They didn't find any difference in the guava root knot nematode population, which is pretty strong evidence that it is one race. Further, when we look at sweet potato varieties from all over the globe that are resistant, they all have the same markers. So it appears to be controlled by the same gene or type of genes. For fusarium, I'm not as experienced with that pathogen. I really just started working with it in the last year or so, or 2 years, I guess. So I couldn't comment on that, but I can look into it and get back to you.
No, that's okay. I can look that up. Thank you.
So we did a survey with sweet potato, fusarium, like years ago, Andrew Crush, remember him? There's a bunch of species that go to sweet potato, that is part of the problem. It's not just potatoes, which you're not surprised because it's happened with every fusarium, every crop ever. That's a big issue. It's like, which of the species do we?
And there is a different race out in California that as far as I know, we're not breeding for, but we may be breeding for their market. So that's something that needs to be considered. And we know that pathogens change.
I have maybe the last question. What is the mechanism of nematode resistance in your advanced selections?
So our initial candidate genes were associated with a hypersensitive response. But my job is to turn those markers into breeding tools. I'm not trying to clone a gene or do a RNA seq or anything like that. However, there are other labs working. Recent graduate from LSU, PhD David Gallo, he just published a paper on that, supporting some of our findings on that candidate gene. What he did was he did a microscope dissection of the fibrous roots of galls, and he looked at nematode penetration and looked at the plant's response. For guava root knot nematode, chromosome 4, his images pretty clearly showed a hypersensitive response occurring. And what a hypersensitive response is the plant recognizes effectors. These are virulence proteins released by the nematode. And if the plant has cultivar resistance, it's going to trigger an immune response and it kills those cells around the nematode. Once she's penetrated in there, she becomes sessile and she can't move. And so when the plant cells die, she dies. Guava is most likely a hypersensitive response. Incognita, different chromosome now, totally different genetic mechanism. I'm not entirely sure. It doesn't seem to be a hypersensitive response. I may be wrong about that though, but that's ongoing research.
Thank you.
I'll give the group one more shot if you would like, but if I don't see any hands, go up quickly. Go ahead.
Yeah, Simon, great talk. I saw in your PCA that very little of the variance was explained by that.
I wasn't asking that one, it is, go ahead.
Explain, why do you think that is? Or I'm not in this field, so I'm not like sure if that expected, and what do you want to do to maybe improve this in the future?
Yeah, so I was just looking at that yesterday in preparation for Craig's question. So, okay, let's take a different example. Modesta, I'm going to use your population. Modesta looked at a biparental population with a huge number of markers, and she did a similar PCA. And her axes explained way more variance than mine. She had a much narrower population composition, and so there's not as much variability. And so one principal component can explain a lot more variance. Here, I have a really, really diverse population. I mean, you can see there's 2 different universities. All that guava resistant stuff is historical. Maybe it's exotic. Maybe it's from the USDA. So it's hard to draw one line through all those individuals when they're very close together. A tighter population structure would probably improve those axes. And we may see that in the NC2 mating design.
And you have fewer markers too.
And we have fewer markers, yes,
Mhm.
exactly.
Well, I think that, let's give him another round. of That says a lot. Well done, Simon, and thank you all for coming. The committee will convene in a couple
Thank you,
minutes
Zoom.
after we wind down. Thanks, Zoomies, for joining us. And, again, well done, Simon. Thank you.